So thank you for calling into the second session, and thank you to Howard and Andrea for asking me to invite speakers to discuss circulating DNA methylation biomarkers for improving the management of prostate cancer patients. The technology in this field has made significant advances over the past 3-4 years, now allowing us to extract high quality data from plasma DNA. And the other two speakers, in addition to me, will be firstly Martin speaking about a very new study on 5-HNC analysis of plasma DNA from prostate cancer. And then last in the session, Francesca De Michelis speaking about using the potential clinical use of methylation biomarkers for diagnosing and identifying neuroendocrine prostate cancer differentiation. And very briefly, to give a high-level perspective, we're here focusing on circulating tumor DNA. There's a number of other tests that can be performed on blood and sources of potential biomarkers such as circulating tumor cells, circulating free RNA, exosomes, and other vesicle types. But most of the work, or the work in today's session, will focus on extracting methylation information from cell-free circulating DNA. So over to Martin with the first talk of the session.
Hi everyone, I'm Martin Sjöström and I'm a postdoc in the lab of Felix Feng at UCSF. And first of all, thank you to the Prostate Cancer Foundation for funding this work and to the session organizers for giving me the opportunity to present some exciting new results from our group. I will show you our ongoing investigation on the novel epigenetic mark 5-hydroxymethylcytosine and how it can be used to track gene activation in advanced prostate cancer. And these are our disclosures. So I would like to start out with asking the question why we are interested in measuring epigenetics in a liquid biopsy. So although cancer is thought of as a genetic disease, the prostate cancer genome typically contains relatively few genomic changes. And with some exceptions, genomic changes are not enough to determine the behavior of an individual tumor or aid in making clinical decisions. It has become increasingly clear that there are profound changes in cancer cells other than to the DNA structure itself. For example, there are changes at the epigenetic and transcriptional levels. And these changes, that are not direct alterations of the DNA sequence, may be much more dynamic and change rapidly, for example, in response to treatment. And many successful biomarkers are based on the gene expression level rather than on the DNA level. However, gene expression analysis currently requires a tissue biopsy, and this is because RNA is quickly degraded in blood and very hard to measure directly from a blood sample. And while some medical centers like UCSF, with the support of the Prostate Cancer Foundation, can obtain a tissue biopsy for metastatic prostate cancer, this is not universally available. So in order to be able to assess this important tumor information in a much wider patient population, there is a need to detect the transcriptional changes from a liquid biopsy. And since epigenetic DNA marks are more stable in blood and regulate gene expression, we believe that epigenetic analysis of cell-free DNA might provide a way of capturing the changes found on the transcriptional level without the need for a tissue biopsy. And I will focus on one type of epigenetic changes, which is the DNA methylation. And I will explain why we think that 5-hydroxymethylcytosine specifically is such an exciting DNA methylation candidate to analyze for this purpose. When we usually talk about DNA methylation, we're referring to the addition of a methyl group to the base cytosine. Unmethylated cytosine is associated with open chromatin, especially at promoters and regions with increased density of cytosine and guanine pairs, the so-called CpG islands. This unmethylated cytosine and open chromatin is associated with gene expression. The methylation of cytosine is performed by a family of enzymes called DNMTs, the DNA methyltransferases, which adds a methyl group to the 5-carbon position of cytosine, which is therefore referred to as 5-MC. This leads to closed chromatin and repressed gene expression. However, it was relatively recently discovered that 5-MC is not the only methylation mark, but that others exist. And one such modification is mediated by the TET family of enzymes, which oxidize the methyl group to a hydroxymethyl group, and the base is then referred to as 5-hydroxymethyl-C or 5-HMC for short. And this mark is only in the order of a couple of percent of the amount of 5-MC, but it is found in gene bodies and on the borders of promoters of highly expressed genes. If this really has a biological role in activating transcription, or if it is a mark of a region undergoing demethylation, is not completely understood, but it is clearly enriched in transcriptionally active regions. And after the oxidation, the cytosine can then be converted back to its unmethylated state again. So this leads us to our main research question. Given that gene expression can be used to create biomarkers, but requires a tissue biopsy, and that 5-HMC is marking active gene expression, we are investigating if 5-HMC can be used as a blood-based marker of gene activation that could be relevant for advanced prostate cancer care. And in order to do this, we have leveraged the very rich data from the Prostate Cancer Foundation and Stand Up to Cancer-funded West Coast Dream Team study. This study was initiated by Eric Small and Owen Whitty and involves many investigators at multiple sites. The study has collected samples from men with metastatic castration-resistant prostate cancer, and many of the genomic studies have been led by Felix Feng and David Quigley at UCSF. And these tissue samples have already been characterized with whole genome sequencing, RNA sequencing, and most recently, whole genome bisulfite sequencing. And this has led to a number of publications on this cohort. So this is a fantastic resource in which we can integrate 5-HMC data as an additional layer of information. And to profile 5-HMC, we have used an optimized pull-down of 5-HMC marked DNA fragments coupled with next-generation sequencing. And this technique identifies regions of increased 5-HMC marking and 5-HMC levels. And this has been done in collaboration with Blue Star Genomics. We have applied this technique to a set of 93 mCRPC tissue samples, and this allows us to perform an integrated landscape analysis of genomic and epigenomic regulation of gene expression, which can then be further tested for the ability to track gene activity in cell-free DNA. So let's move over to some results. And first, I would like to give you a quick glimpse of the landscape part of this study. In addition to the West Coast Dream Team cohort, we have also worked together with the Canadian Prostate Cancer Group and Hansen He at the University of Toronto and profiled an additional 55 localized prostate cancer tissue samples. This allowed us to start out with a genome-wide global analysis of 5-HMC changes. We assessed differential 5-HMC markings between localized, hormone-sensitive, and metastatic castration-resistant prostate cancer in regions identified to have 5-HMC modifications. And in this figure, we plotted the most significant changes out over each chromosome. Red in this figure means that the locus is gaining 5-HMC in mCRPC, and blue that 5-HMC is lost in mCRPC. And this analysis readily identified increased 5-HMC signal in known drivers of mCRPC, such as AR, the PVT1 and MYC locus, and FOXA1. Interestingly, it also found loss of known tumor suppressor genes such as RB1. But clearly, many more regions are marked by increase or decrease in 5-HMC throughout disease progression. And if we map the top upregulated regions in mCRPC to the nearest gene and regulatory regions, a pathway analysis identified high enrichment for transcriptional programs associated with development and dedifferentiation. And I think this is very exciting, as a recent study led by the Mark Pomerantz, Wilbert Swart, and Matthew Friedman groups reported similar findings for epigenetic reprogramming of the AR, FOXA1, and HOXB13 cystrones. So in summary, this technique has the potential to track gene activation and activity loss of drivers of mCRPC. But since this is a 15-minute talk, I will focus the rest of this presentation on the association between 5-HMC and gene expression and potential biomarker applications in cell-free DNA. But anyone interested in collaboration is very welcome to contact the group. So the next set of analysis is aimed at moving from the landscape of 5-HMC to biomarker potential. First, we compared how well 5-MC in promoters correlate with gene expression. And remember, this is the traditional DNA methylation mark that is associated with repressed gene expression. And then we calculated how well 5-HMC in gene bodies, this novel marker associated with active transcription, how well that correlated with gene expression. And promoter methylation was negatively correlated, as expected, while 5-HMC in gene bodies had a considerably stronger positive correlation with gene expression. And this is consistent with a model where promoter methylation is more of an on-off switch of gene expression, while 5-HMC is better for measuring fine-tuning of transcriptional activity. We then asked for which genes the correlation was strongest, and the gene set enrichment analysis showed that the androgen response genes was clearly the most enriched pathway, suggesting that 5-HMC marking of actively transcribed genes is done in a disease-specific manner. We then integrated the 5-HMC data with all other data available for the West Coast Dream Team cohort, such as copy number, promoter methylation, structural variants, and single nucleotide variants. And this was to create a complete model of gene expression. 5-HMC was a strong independent predictor of gene expression for most protein-coding genes, and the strongest added value was seen for the androgen response genes. The bar plot to the right in this slide shows models with or without 5-HMC in predicting gene expression. The bar height is the median gene expression explained by the model, and the orange bar is the model without 5-HMC, while the blue bar is the model with 5-HMC. This shows a significant increase in gene expression prediction by 5-HMC. And to summarize this analysis, we show that 5-HMC significantly and independently predicts gene expression, and that the prediction is strongest for the AR response genes. And together, this highlights that 5-HMC can be used to detect gene activation in a disease-specific manner, and that it provides information beyond other types of data. And now I will continue with the analysis of 5-HMC in cell-free DNA. To do this, we have collaborated with Alex Wyatt and colleagues at the University of British Columbia and profiled a set of 64 cell-free DNA samples that are from men with mCRPC before start of first-line novel generation androgen signaling inhibitors. So just to remind you, cell-free DNA found in the circulation can come either from the tumor cells or from normal cells, most commonly from leukocytes. And many publications have described that the amount of tumor DNA in a cell-free DNA sample is associated with disease burden and is prognostic. And methylation is tissue and cancer specific, and it has also been shown by, for example, the groups of Gertrude Tard and Francesca De Michelis that methylation patterns in cell-free DNA can be used to estimate the amount of prostate cancer in a cell-free DNA sample. So we therefore hypothesized that also 5-HMC patterns could be used to estimate cancer content in a liquid biopsy. And we used our tissue data, and we created a 5-HMC classifier that could predict the amount of prostate cancer in a given sample. And the 5-HMC predicted prostate cancer content in cell-free DNA was split into tertiles of low, medium, and high. And the amount of prostate cancer in the sample was highly prognostic for overall survival in this cohort. So to summarize this analysis, 5-HMC levels alone can estimate cancer content in the sample, which is in turn prognostic and opens up for use of 5-HMC to, for example, monitoring treatment response. In this next analysis, we wanted to test also in cell-free DNA the finding from the tissue analysis that 5-HMC detects gain and loss of common oncogenes and tumor suppressor genes during the progression to mCRPC. We therefore selected a set of common key drivers, both oncogenes and tumor suppressor genes. And first, we confirmed that the 5-HMC levels were associated with the results from targeted sequencing that was previously performed on these samples. And very encouraging, the 5-HMC levels were in accordance with called alterations, both for genes for which copy number is the main way of activation or loss, and for genes that are activated or altered by other mechanisms. We then tested if the number of oncogenes with gained activity and tumor suppressor with loss of activity, determined by 5-HMC levels, was associated with prognosis. And high activity of oncogenes was defined as the highest quartile of 5-HMC levels, and low activity of tumor suppressor genes was defined as the lowest quartile of 5-HMC. The number of genes with gain or loss of activity determined this way by 5-HMC sequencing was strongly prognostic in cell-free DNA, and importantly, it remained prognostic after adjusting for the amount of prostate cancer in the sample. So in summary, we can demonstrate that 5-HMC sequencing of cell-free DNA can be used to infer gain or loss of common oncogenes and tumor suppressor genes in mCRPC. And finally, after demonstrating that 5-HMC can be used to detect gain or loss of some of the most commonly altered genes in prostate cancer, We wanted to explore the opportunities of this genome-wide assay that provides 5-HMC levels for all genes. And we have previously shown, together with the lead authors Lee Ellis and David Labby, that the combination of high expression of the genes TOP2A and EZH2 identifies an aggressive subgroup of prostate cancer. We tested if this classification of mCRPC is possible also in cell-free DNA using 5-HMC levels. And this was highly prognostic for overall survival, where high levels of 5-HMC of both TOP2A and EZH2 together conferred the worst prognosis. And this result is highly significant also after adjusting for amount of prostate cancer in the sample, and neither TOP2A nor EZH2 are commonly copy number altered. So this analysis highlights the potential of 5-HMC analysis to go beyond what can be detected by, for example, targeted sequencing of cell-free DNA. And to wrap things up, I will summarize some main conclusions so far of this ongoing study. I have showed you that 5-HMC is a disease-specific marker of gene activation in advanced prostate cancer. Further, 5-HMC is associated with gene expression, and this association remains after adjusting for other types of data. The association is strongest for the AR response genes, suggesting a disease-specific marking of 5-HMC. And 5-HMC patterns in cell-free DNA accurately detects the amount of prostate cancer in the sample, which is strongly prognostic. And 5-HMC in cell-free DNA can infer activity of key oncogenes and tumor suppressor genes, which is highly prognostic and provides information beyond the amount of prostate cancer in the sample. And finally, 5-HMC is a genome-wide assay and can infer activity of genes not normally part of targeted cell-free DNA panels, such as TOP2A and EZH2, which identifies an aggressive subgroup of prostate cancer. And ongoing work is aimed at optimizing and validating 5-HMC profiles in cell-free DNA for prognosis and treatment response in larger cohorts. And I would like to conclude by first and foremost thank all the patients who have donated samples to this study. None of the research would have been possible without you. And this study is really a collaborative project. First and foremost, I'm very grateful for the leadership and mentorship of Felix Feng, who leads this study. And further, the entire team at the Feng Lab and UCSF have been a fantastic support, and I would especially like to mention David Quigley, who oversees much of the computational work, and Eric Small, who provides tremendous clinical and translational insight. In addition, I would like to thank Sam Levi and our collaborators at Blue Star Genomics, who have optimized the 5-HMC sequencing technology. I would also like to highlight the West Coast Stream team and collaborators at other institutions, especially George Tsao at University of Wisconsin and the teams at University of British Columbia with Alexander Wyatt and colleagues, and at the University of Toronto and Hansen He. And finally, I would like to acknowledge all the funding agencies and especially thank the Prostate Cancer Foundation who make this kind of collaborative work possible. Thank you so much, and I'm looking forward to the discussion.
So hello everyone, the second talk in this session, that's my title. I'll start with my disclosure slide, which is listed here. So in the next 15 minutes, I'll present an experiment we performed, and that was published earlier this year in the Journal of Clinical Investigation. We had been studying the genomic aberrations in plasma DNA for, I guess, several years. And in a number of large cohorts, we had studied gene aberrations, and there was additional DNA that had been extracted and that was available. And we were interested in maximizing the information extracted from this, and specifically understanding changes in methylation. So from these samples, which we either had done or performed genomic profiling, we also did bisulfite treatment and then high coverage targeted next generation sequencing on DNA. And you can see the details on the protocol in the publication, which is referenced at the bottom. We specifically chose 25 metastatic CRPC patients who are all starting either abiraterone or enzalutamide, where we had pre-treatment samples and progression samples. And because we expected the methylome to differ by tumor content, we were careful to select a range or samples with a range of different tumor fractions. as you can see here, and relatively equally balanced for men who were pre-docetaxel versus post-docetaxel. And the first experiment we did to perform a dimension reduction of the data was principal component analysis. And what was striking was that a high proportion of the variance, more than 40% of the variance in methylation status in the set of samples was attributed to one component. And we went on to investigate principal component further and appreciated that this was strongly correlated with tumor fraction. So what proportion of the DNA arose from tumor? And that tumor fraction had been defined using a genomic test. And this really was an orthogonal test that we couldn't see any reason for the methylation event to be biased by the previous genomic assessment. We also controlled to evaluate whether regions involved in this principal component analysis were enriched for specific copy number changes, and we didn't see that. We proceeded to choose the top ranked segments within our signature that show the highest correlation with tumor fraction. You can see them here split by the hypermethylated group and the hypomethylated group, relatively equally split. And we call this signature CT-MET-SIG for circulating tumor methylation signature. We're then interested in evaluating this further. And we performed enrichment analysis and found that in the hypermethylated segments, there was significant enrichment for components of the polycomb repressor complex 2 and targets of this complex, as you can see here, and with significant overlap for enriched genes that were hypermethylated in our signature being targets of PRC2. We then hypothesized that the components in the signature would include methylation events that were both normal prostate epithelium specific and cancer specific. And to test this hypothesis, we looked at the Gaussian distribution in LNCAP cell lines, in a number of CRPC metastases, in control healthy volunteer white blood cell DNA, and then in a number of control normal prostate samples. And you can see here on the left side, regions, which was about approximately half of the regions we selected for the signature are cancer specific, and about half are shared both in the cancer samples and the normal prostate epithelium. And this all was making sense to us that there are methylation events in prostate cancer epithelium that are unchanged compared to normal prostate epithelium, and also events that are specific to cancer. It's also important to know that what we're identifying here are segments in the genome, and our panel was a targeted panel that included pan-genome CPGs. We're identifying regions that show the highest variance, and this differs from other experiments that have compared normal prostate cancer with, prostate cancer cells with normal prostate. And for example, GSD-Pi, which is a region which repeatedly shows enrichment for hypermethylation in cancer cells, doesn't show that high level of variance in our experiment. And we find that to be explained by hypermethylation or levels of hypermethylation in white blood cells that make that specific gene or segments around that gene not useful for quantitating the prostate fraction, the prostate tumor fraction. When we originally designed the experiment, we were also interested in understanding methylation signatures that define biological subgroups. And we went back to our original principal component analysis, and there were two other components that showed significant enrichment. PC2, or the second principal component, was driven by a very small number of samples. So we focused on principal component 3, which showed a more balanced distribution across our cohort. And this specific, we selected again, same as previously, the 1,000 segments that show the highest correlation, and proceeded to investigate this further. And interestingly, well, firstly, we performed enrichment analysis and did not identify enrichment for any specific pathway. But we then noted that the hypomethylated regions were enriched for proximity to AR binding sites, and suggesting a signal in a subgroup of our metastatic CRPC cases that were characterized by hypomethylation events in these regions. And a recent publication from Matthew Pomerantz, published in Nature Genetics, shows a similar result in an extensive study of the epigenome in prostate, normal prostates, prostate tumors, and metastatic CRPC samples with similarly hypomethylation events in AR binding sites. Now, what we find is a clear challenge in studying the methylation in this way to define biological subgroups is that the signal is challenging to dissect in cases with a low tumor fraction. And that's ongoing work in my lab to define the computational processes. But I suspect implementing a study like this in cases where the fraction is low, less than 5%, as we would often see at earlier stages of the disease, introduces a number of challenges. But what we've become particularly interested in is using this methylome signature to track tumor DNA and achieve high sensitivity. And there's a number of opportunities that methylation analysis allow that could be superior to genomic analysis. And certainly key amongst those is that we are measuring a large number of methylation events, not a single genomic change. So we hypothesize that would improve the sensitivity for detecting tumor at low fractions. And as I said before, I said earlier in my talk, we're also detecting, or a significant component of this signature is driven by changes in normal prostate epithelium, which we hypothesize will be present throughout all cells, all cancer cells, and not subject to intratumor heterogeneity, which remains a particular challenge when tracking a single genomic aberration. So finish off with acknowledgements. This is my lab group that did a lot of the work, driven by Andrew Wu, who's a very talented PhD student, and Daniel and Paolo, senior post-PhD scientists in my group, and a number of collaborators who were instrumental to making this work possible. So look forward to discussing this data in the Q&A later in this session.
So good morning to the PCF community. I'm very happy to have this opportunity. So loss of androgen receptor signal independence occurs in up to 20% of advanced treatment-resistant prostate cancer, and multiple mechanisms and manifestations have been reported, including the transformation of adenocarcinoma histology to a castration-resistant neuroendocrine prostate cancer. Now, the prognosis of CRPC&E is really poor, and this is due to lack of efficient treatment and also at times too late diagnosis. And the latter currently occurs through a metastatic tissue biopsy. Now, this is obviously suboptimal for the patient because it's an invasive assay, and it's also tough to diagnose at times due to morphological heterogeneity. So in this context, the aid of a liquid biopsy assay would be obviously of importance. Over the past few years, in collaboration with Dr. Bertrand, we and others working on tissue-based data reported on peculiar genomic lesions of CRPC&E, and then also to a massive epigenetic reprogramming in it. So we thought that to really test the feasibility of a liquid biopsy assay in this setting, we could exploit a tissue-informed study with matched liquid biopsy, so that we could see how well this tissue signal would reflect in the circulation of the patient. And we had an extreme design with men with high metastatic burden. And nicely, we had multiple metastatic tissue biopsies or serial liquid biopsies for a subset of patients. And to really embrace the genomics and the epigenetics, we ran whole exome sequencing on the whole cohort and also whole genome bisulfite sequencing on a subset of them. So the first question that we really wanted to ask by leveraging the metastatic tissue data was to measure what is the similarity of metastatic biopsies at different sites. And this is due to the fact that we know any disease has preferential seeding sites for metastasis. As you can see here, there is no major difference in the similarity of intra and inter-patient metastatic genomic profile when we look at the same sites or different sites. And also when we looked at the circulation cell-free DNA tumor content, we did not observe an association with the site of metastasis or the number of metastases. And these two observations suggested that, in fact, we could use the metastatic site tissue biopsy to inform our study and really look at the differences that are proper to the two subgroups and not strictly related to the site of a met. And so we charted the distribution of the similarities intra-patient between all the metastases for each individual. And what we saw was a marked increased homogeneity or concordance in the NE subgroup versus the adenocarcinoma. And when then we brought the liquid biopsy into the game, we observed the same signal whereby the NE patient had a much higher level of homogeneity. And this suggests that in principle, one could also think in terms of entropy-based markers within a liquid biopsy assay to distinguish what is a neuroendocrine disease versus an adenocarcinoma. Now, this is the ultimate example of neuroendocrine homogeneity, where we delve deeper into the data and look at allele-specific information. So to the left, you see the plasma data. To the right, the plasma data. To the left, the lymph node metastasis. And by looking at the data at the allele level, you see that basically the two spectra are basically identical. Whereas if we looked into other patients, we see that there is a lot of similarity between a liver met and the matched plasma, but also some subclones that bring different signal that here is highlighted in blue. So what does that translate into when we looked at methylation? So first of all, we were happy to see that by using few tenths of methylation sites or CpG sites, selected in tissue, we could measure the quantity of circulating tumor from methylation data in the plasma. This is on the right side. And then when we look at overall the differential methylation signal, comparing the tissue on the x-axis and the plasma on the y-axis, we saw overall a good concordance, suggesting that what we learned from the tissue metastasis is reflected in the circulation. And this is also true when we then focus on very specific markers, including NE markers, and also SPDEF, which is hypermethylated in NE and overexpressed in adenocarcinoma. So very neatly, although we use different platforms, the tissue and the plasma methylation data were highly concordant. This is the most beautiful piece of data in my view when we looked at the patient level. This is all about two sets of differentially methylated regions, here denoted with DMRs. The first one, which is the first column, relates to localized disease versus normal prostate tissue. And then the second column will be NE versus adeno. And here we are looking at two patients. The top one is an adeno, the bottom one is an NE patient. And you can see that for the first set of DMRs, we see signal both on plasma and tissue for both of them. But then when we looked into the plasma data of both samples, we only see concordant signal, or actually we only see signal for the NE patient and not for the top one, suggesting that in fact certain regions are really specific for NE and are mimicked in the circulation. Another layer of information is that when we looked at a third patient here, which has mixed features, we could see an intermediate signal. So all in all, this suggests that, in fact, methylation in the circulation is reflective of neuroendocrine disease, and we came up with a partial classifier on epigenetic first that not only was able to discriminate NE from adeno in our data set, but also an independent data set published, plasma data set published from Goddard-Vicious in 2018. Very importantly, here you see on the very left the PBMC signal that we used at the level of DNA methylation to increase the ratio of signal to noise. And then we combined the genomic and the epigenetic in a score that was in fact very simple, with very few genomic features and few tenths of hyper and hypomethylated differential region. And so this together speaks towards the real feasibility of a classifier for the circulation. 2 anecdotal sample of multi-sample patients. This is a plasma sample at time of CRPC adeno with a lymph node and a bone metastasis that displayed a genomic cell-free DNA profile that was most similar to the CRPC and NE liver met that was detected 3 months later at time of progression on abiraterone. And this is another example of spatially distinct metastasis where, again, the plasma sample was more reflective of the liver met with respect to the other ones, suggesting that maybe liver metastases do release more efficiently in the bloodstream. So the summary of this part of the study that was published early this year is that, in fact, the feasibility was successful in that circulating tumor DNA does capture both genomics and epigenetic features of NE disease, and then also that NE disease in the late side of the spectrum is more homogeneous than adenos. Now, when we deal with liquid biopsy in the setting of CRPC in general, not only for NE, we have to remember that we are in a very difficult setting because the genomics of the disease here is multi-layered and offer polyploidy. And although polyploidy does not associate with worse prognosis or a relapse-free interval, is really enriched in late disease versus localized, and that brings into the play multiple layers of genomic states that also affect the ability to detect methylation. Another piece of information is that, in fact, the ability or so the performance of liquid biopsy assay not only depend on the gene region, but also depend on the patient and also depend on the tumor content at each specific time point. This is an exercise that we ran together with the PCF Select team. And if you only focus on the left side, here we see one patient. The red bars indicate the ability to detect a lesion for that specific gene for that patient, while we dilute synthetically the tumor content by adding reads from the matched germline. And as you can see from where the line stops, for certain genes, we have ability to go down to 6% of tumor content, but for other genes, we do not. And so there is a gene specificity in our performance measure. Now, how can we use this whole information to make a better liquid biopsy assay that eventually could hopefully be used also in the clinic? So we did invest in the past few years on a better way to detect differential methylated signal. This is based on AUC measures. We developed it, we applied to the whole TCGA and came up with catalogs that more importantly showed a nice functional annotation with respect to other compendia that are published. And not only we obviously detected hyper and hypomethylation, but also very nice super long stretches of hypomethylation regions that we called hypoways. When we applied this to all the epigenetic data that we could get at the beginning of the year from prostate cancer, what we saw, and this is on the left side, is that differential methylation signal between CRPC adeno and localized disease is modest compared to what we see between NE and adeno. And then by looking at the overall underexpression of genes, we saw that specifically focusing on upregulation at promoter, we see the expected correlation, suggesting that these DMRs are nicely, let's say, functional and are mimicking something that happens in the tumor. A second piece of information was inspired by this very beautiful paper from Park and al., published in Science 2018, where the WITA group used the ATAC-seq data to study accessibility of the chromatin. And they did that by using normal epithelial prostate cells versus neuroendocrine-like transformed cells. So what we have done was focusing on differentially methylated regions between CRPC-NE and benign prostate tissue. And then we run transcription factor enrichment analysis in the DMRs. And then we compared the signal against what was reported in the WITA paper. And as you can see on the right, there is a very nice concordance between these two measures that suggests that differentially methylated signal can be used as proxy of chromatin accessibility. So putting together this information, we are really enthusiastic in developing a targeted assay based on DNA methylation that is differential. And what we are using are the dynamic patterns of all these regions across the disease states in prostate cancer. And here you see all these facets. Each of them, you can see, has a different profile. And the selection for the final assay will come from this landscape. And we'll also be informed on the methylation signal that come from other sources that can release in the circulation. This is again to boost our signal to noise ratio. So very briefly, I like to show you that when we take DMRs that we had selected by using all these steps that I mentioned, and we project them on a totally independent data set that just became available. This is 100 metastatic CRPC whole genome sulfide sequencing data from the West Coast Dream Team. Zhao is the first author of the paper. And we use the DMRs that we selected. We can very nicely segregate the whole cohort. But what is more strikingly and more interestingly, even though it does not project here on the slide, you can see from the slopes of these lines that represent the signal of hyper, hypo, and hypoways, that there is a really strong correlation of the DMRs in the two cohorts. So we do believe that we can get towards an assay that can be used for CRPC-NE. Obviously, we needed to move to a little bit more in the middle of the spectrum and not only using the very extreme as we have done so far. We see a great value in informing the selection of this assay from tissue-based data, especially for the NE disease that is not that common, and so we cannot only rely on plasma cohorts. And we also think it's so important to be able to share data to inform, train, test, and validate any type of assay that we develop. We are obviously embracing open questions that we have not even touched yet. One is, will we be able to detect any predisposing features in earlier stage disease? And will a similar approach be used possibly for patient selection for trials in the future? So with that, I like to conclude by recognizing especially the work of Gianmarco Franceschini, who is a brilliant PhD student who developed the majority of the unpublished data that I presented. He's been working in collaboration with Matteo Benelli, who now is leading his independent group in Tuscany. I like to thank the PCF Select team, and then very importantly, the funding agencies that supported this work over the years. So the Prostate Cancer Foundation, the Movember, and then more recently, the Weill Cornell Medicine SPORE that is supporting this beautiful project that I'm co-leading with Dr. Beltran and Dr. Tagawa. So with that, I thank you for your attention, and I'm really looking forward for the discussion session of this panel. Thank you.
Hello, everyone. Thank you for calling in, and thank you for posting so many questions. We're going to have a really good debate for the next 14 minutes. My name is Derek Hathard. I'm a medical oncologist in UCL London. So the first question for Martin, it's from David Vanderweel from Northwestern University. I'll read it out. Is there a novel 5-HMC signature that is prognostic, aside from looking at 5-HMC at known oncogene tumor suppressor genes? And for the prognostic signatures you show, do they remain prognostic when accounting for other known clinical variables? So Martin?
Yes, thank you very much for that question. I think it's an excellent question, and that is definitely what we're ultimately trying to do, to develop novel prognostic and treatment predictive signatures, and also port over already known gene expression signatures to cell-free DNA. So in this set of 64 samples, we have started with trying looking for known biology and get a positive control and see that we find the expected biology we're looking for. I think it's hard in 64 samples to really train and validate a novel signature, but that's definitely something we want to do in the future, and we are analyzing more samples.
The next question from Andy Armstrong, I think was initially maybe directed to Martin, as that came through in Martin's talk, but very relevant to Francesca's as well, so I'll read it out. Does 5MC versus 5HMC expression at the AR locus explain the loss of AR activity in NEPC and small cell patients? And has this been tested in cell line models like PC3 cells? So Francesca, you've done extensive work on 5MC analysis in tissue and ctDNA. Can you comment on the changes at the AR and how those relate to the neuroendocrine phenotype?
So in fact, thank you for that. He'll have a lot to say on it. So in our experience, we haven't really noticed a significant signal change in the surrounding of AR in terms of methylation. And that is something that surprised us, because when we saw reports from others, we obviously realized that could be one of the cause of the signal, and may be related to the differentiation of the cells. That said, because this is gaining attention, it's something that definitely needs to be investigated, and we'll probably go back to the tissue data rather than insisting on what we can do in cell-free DNA.
Martin, can you hear us? And
yeah, I can hear you.
Okay. So you were rightly explaining that in a relatively small cohort of 60 or so patients, there was a limit to the prognostic and signature model development. and the testing of that with other clinical variables. Was there anything you wanted to add to that question?
No, I think
Or
that was
your answer?
basically the answer. And I think just also want to say that I think the potential of this assay is that we get genome-wide estimates for activity levels of the genes. So I think the strength would be to create these types of multi-gene signatures.
And so going back to the question Francesca gave her reply to, and you talked about 5-HMC and the changes around the AR locus. Do you think, or do you hypothesize those would explain the loss of AR activity in the neuroendocrine phenotype?
So we definitely see that they correlate with the neuroendocrine phenotype. And we have a poster as well presented by David Quigley looking more into 5-HMC changes between small cell and adenocarcinomas. So we definitely see a big change in 5-HMC levels. So yeah, it correlates. I don't think we can say now if it's causative or not, but there's definitely a big change in 5-HMC levels.
I have a question for both of you, and it's a challenge my group have been, I guess, struggled with. What do you think is the lower limit of tumor fraction that would allow you to make these biological inferences or detect a tumor-specific signature? In Francesca's study related to neuroendocrine tumor changes. But I suspect this is going to be challenging in earlier disease where the fraction is low. What's your opinion? Maybe Martin first and then Francesca? So I think the question about the tumor-specific signature, an individual cancer-specific signature.
Yeah, I think you're definitely right that the CD fraction is what's limiting the use of some of these analysis. I think it's different what you're trying to do with the signature. If you're trying to measure the expression levels, for example, between different cancers, or if the goal is to try to find a cancer-specific signature versus normal cells. So I didn't have time to mention that in the presentation, but for most studies, the most studies on 5-HMC have been in the diagnostic setting, comparing healthy control versus cancer patients. And there are some preliminary, very exciting data that it appears to be really good at picking up the cancer-specific signal. And there was a preprint just a couple of months ago looking also in prostate cancer with some exciting results. So, yeah.
Okay, Francesca?
So, in our experience, I think so far, we can say that detecting methylation change in vision, it's probably a little bit easier than detecting copy number changes. And as you know, because you're part of a PCF select team, when we look at genomics, we can't really go lower than 5% tumor content if you really want to assess a whole panel of genes. Now, it's very different if we know that one gene is aberrant and we have serial time points and then we want to follow, because then we have some extra trick that we can use in analyzing the data. Looking for DNA methylation changes, and we can deal with longer stretches of DNA and counting on many data points that at times really make this signal or the signatures that we are looking for, I think we'll probably be able to go a little bit lower. Now, that said, I cannot say how low because we didn't do an extensive study or an extensive enough study to have a whole spectrum of that's definitely something that needs to be done and maybe can be done by syntactic dilution, so in silico dilutions from generated data, or also may be done using cell lines and intervening with normal DNA for the methylation detection. But it's definitely something that we as a community, we all should try to push, because probably as the signatures do include multiple sites or multiple differentially methylated regions, lower in terms of tumor content.
Okay. Yeah, and I think that ties into a later question we were asked about the utility in active surveillance. So from my experience, I think these type of studies could be very sensitive for detecting tumor content and detecting tumor to be present. But do you think there's also an opportunity for interrogating gene expression or potentially neuroendocrine-specific signatures at the low levels of tumor content in earlier disease settings. Does that make sense? Francesca first, then maybe Martin.
Sure. Impossible to say at this very moment for me, based on the data that we have. I think that if we go earlier into the disease, we should probably look at a combination of assays. One option would be to maybe bring in extracellular vesicles and look at the transcript that is carried in these vesicles, say as a cargo, and then maybe in there detecting point mutations that are prostate cancer related could be just digital evidence of yes, there is cancer or no. I don't think we can do it with genomics only. And I don't know about methylation, but definitely something to investigate. Probably, in that disease state, I would say that we can go more with evidence of cancer. Yes, if there is no evidence, it's hard to be sure that there is nothing.
So, a question for Martin from Vassie and Johns Hopkins. 5-HMC has often been thought to be simply an intermediate state on the way to demethylation of 5-MC. However, you're showing associations with 5-HMC in the gene body and increased expression. So what's your thoughts on that? Did you find any specific transcription factors or potential readers of the 5-HMC mark that could support it?
Yeah, I think that's a very, very good question. And I don't think that the knowledge is there yet to say exactly how 5-HMC associates or promotes transcription. There are exciting studies showing perhaps that there are readers or proteins that can bind to 5-HMC specifically, but that's nothing we have been able to test in our data.
Okay. And so an interesting question, Hansen He. Can methylation signal from tumor microenvironment be detected in plasma samples? Hansen He in UHN. I've not thought about this before. I'm not sure how we'd interrogate it, but Martin first, any ideas?
Yeah, I haven't thought much about that either. I mean, I guess it depends on how you could try to tease out what's coming from the leukocytes or from other parts of the normal DNA, or potentially from the microenvironment. But I don't think I have a good answer to exactly how I would analyze that right now.
Francesca?
Yeah, I never thought about that either. I guess, one would have to study methylation of nearby cells in tumor tissue first, see what is the level of differential signal, if there is any field effect, as it was proposed, either related to methylation, and then if there is one, then probably that can be propagated in the plasma and be detected. But then how you can dissect that from what comes from the stroma and what comes from the tumor cells, and that's something that I don't think can be sorted out, basically.
I think that's fascinating.
Yeah.
A future PCF Challenge Award, that's going to require some deep thought. So a question for Francesca from Dolores Di Vizio. You mentioned that there is some gene specificity in the depth of the signal. What do you think is that, and what genes can be better characterized?
I'm not sure I exactly got the question. What I was intending referring to the vesicles is that if we see point mutations that are specific to prostate cancer that are in the RNA from vesicles, then we can say very likely those vesicles come from cancer, from prostate cancer, and therefore there is material in the circulation. So I was not thinking about anything specific, but just what we know is the landscape of genetics in coding regions for prostate cancer.
So our time is up. I really flew by. I hope that was interesting for everyone. And can you please go back to the virtual lobby to join the next session. Thank you all for listening.